Welcome everyone. Thank you for showing interest to our project, which is electroencephalography neurofeedback for decoding and modulating human emotions. The authors of this research are Sarah Zahmi, Bashar Al-Yamahi, Mithil Yamahi, Maryam Shamsi, and Dr. Abdul Qadir Balqasem. We are from United Arab Emirates University. In this presentation, we're going to start with introduction, and then we will discuss the motivation behind this research, and then we will jump to the methods and materials, where we will discuss how the system works, design and architecture, implementation requirements, and implementation and testing. Then we will discuss the obtained results, and lastly, we will end up with the conclusion.
A human brain consists of millions of neurons communicating with each other using electrical charges and chemical ions. BCI technology is used to measure the electrical brain activities, which provides different techniques for recording, visualizing, and analyzing these activities. Furthermore, the brain is responsible for the emotions that a human can experience. Therefore, these emotions are playing an important role in human health, which is associated with the human interaction with the surrounding environment and decision making. In this research, we introduced a neurofeedback system that can decode and modulate human emotions using non-invasive EEG technique.
The motivation. Psychiatrists used to treat psychiatric patients using medication, sedatives, or cognitive behavioral therapy, which may have side effects that negatively affect human health in the long run, don't show good results, or some patients refuse the therapy. Furthermore, UAE supports the ideas and projects that spread the awareness of taking care of the overall health according to the National UAE Strategy for Well-Being 2031, which aims to make the UAE world leader in quality of life. Our proposed system for decoding and modulating human emotions using non-invasive BCI technology is an effective therapeutic system that will enhance an individual's ability to modulate his or her brain activity to treat mental illnesses in healthier manner with the least side effects. Methods and materials. Here we will come to the methods and materials, where we will talk about how the system works, the design and the architecture of the system, implementation requirement, and implementation and testing. How the system works. The aim of this study is divided into two parts: decoding human emotions using EEG technique to diagnose the emotional state of the person, and training the person to regulate his brain activity using the neurofeedback techniques as a treatment to enhance emotional performance. Neurofeedback actually is a biofeedback that delivers audio and visual feedback representing positive or negative feedback for desirable and undesirable brain activities. To modulate the brain activity of a psychiatric patient and heal chronic sadness, depression, or anxiety using the EEG neurofeedback system, the patient must wear an EEG headset consisting of electrodes connected wirelessly to a PC and set in front of a screen. The neurofeedback sessions last for 20 minutes, and the system decodes the patient's emotions every 60 seconds, and visual and auditory feedback are generated on a screen using the decoded emotions. The feedback consists of motivational scenes, music, and psychotherapy textual instructions to train the patient to keep thinking positively. Design and architecture. Secondly, we come to illustrate the design and the architecture. The system must be implemented in both offline and online modes. In the offline mode, We decode human emotions by collecting EEG data for happiness, sadness, and neutral emotions from many participants. These collected EEG data are pre-processed and then feed into SVM model that can predict the type of human emotion from any incoming EEG data. In the online mode, the second objective of modulating human emotions via neurofeedback sessions is considered. Here, a patient who is suffering from chronic sadness is asked to wear an EEG headset and sit in front of the screen. The system records his brain activity in real time and compares it with the previously developed SVM model to decode the emotions that the patient is feeling in that instant. From the decoded emotion, visual and auditory feedback is generated by the system to train the patient to regulate his brain to keep thinking positively.
Thirdly, we come to illustrate the implementation requirement. There are important requirements to consider when conducting the system in the offline and online modes. The first requirement is an implementation requirement consisting of 3 parameters. The first parameter is identifying the target's brain region. The human brain is divided into 2 hemispheres, each hemisphere into 4 lobes: frontal, parietal, temporal, and occipital lobes. Each lobe is responsible for a specific function for controlling and coordinating the human body. In this study, we focused on studying the emotions from all lobes in both hemispheres of the brain. The second implementation requirement parameter is to determine a method for measuring brain activity. The electrical brain activity is measured invasively and non-invasively using different techniques such as EEG, fMRI, and MEG. In this study, the electrical brain activity was measured using non-invasive EEG headset. The last implementation parameter determines what feedback to present to the patients. The effectiveness of neurofeedback sessions is determined by the type of feedback presented to the patients. However, there is no specific proof for any feedback that can show a better influence on the participant. The feedback could be visual or auditory or both. In this study, we used the feedback that displays multiple motivational video clips with music, scenes, and psychotherapy textual instructions that can differ based on the detected emotions. The second requirement is the system requirement, which explains how the system must function in terms of hardware and software. For the former, the system acquires brain activity using a unicorn hybrid black EEG headset that is connected wirelessly to a laptop and store the acquired data in a CSV file. For the latter, the EEG data were acquired using a unicorn API and then proceed and analyzed. For the latter, the EEG data were acquired using a unicorn API and then processed and analyzed using Python to be fed to a machine learning model. Note that the system must be able to process and analyze any incoming EEG data in real time to generate visual and auditory feedback.
Fourth, we come to illustrate the implementation and testing. The introduced system requires implementing multiple steps in sequence to make the system succeed and achieve the main objectives. The following figure illustrates the sequence of the required steps to be implemented. First step is the experimental paradigm, where we have asked 12 female participants to record their brain activity while they are watching videos that evoke happiness, sadness, and neutral emotions. This because to develop a system capable of distinguishing happiness, sadness, and neutral emotions from the brain activity, we need to collect EEG data or the corresponding emotions. The following figure illustrates the protocol followed by the participants during recording their brain activity. The second step is EEG signal acquisition. The brain activity of each participant were collected using Unicorn EEG headset that is connected wirelessly to the PC with a 24-bit resolution and 250 Hz sampling frequency. The third step is pre-processing the collected EEG data. In this step, we have applied independent component analysis to remove the eye blinking artifacts from the data, as illustrated in the following figure. Furthermore, 50 Hz notch filter was used to remove the power line artifact, and also bandpass filter were used to extract alpha and beta bands from the EEG data because they represent the emotional process of the brain better than other frequency bands. The fourth step is classification. The pre-processed EEG data were fed to support vector machine learning model with radial basis function kernel to categorize them into three classes: happiness, sadness, and neutral. The last step is the user training, where the neurofeedback session will start, and the psychiatric patient will wear the EEG headset and sit in front of the screen, and the system will start decoding the emotions every 60 seconds and will generate visual and auditory feedbacks to train him how to think positively.
Move on to the results that we obtained from this study. On the screen in front of you, there is a table and a figure. The table shows the values of the evaluation metrics used to evaluate the performance of the CVM model. And the figure shows the confusion matrix that gives summary of the prediction results from the classification. These measurements show satisfactory outcome. That means that the system can decode the correct emotion after 2 or 3 trials only. This little variety in prediction of the correct type of emotion from the first trial occurs because of 2 possibilities. The first possibility is the length of each video for the brain activity of each emotion, because it was recorded for 60 seconds, in which the emotions of each participant fluctuated, and thus it is inaccurate enough to study the happiness, sadness, and neutral emotions. To be more accurate, the brain activity should be recorded for a very short duration, representing only extreme evoked emotion, while participants watches the stimulus. The second possibility is that not all participants felt the same emotions for displayed happiness, sadness, and neutral videos, thereby causing variations in the results. The neurofeedback session will show a noticeable improvement in the mental health of the patient within 10 to 20 sessions only. And for the best results, the patient must perform between 30 and 40 sessions. To evaluate this improvement, the system during the last session decode neutral and happy emotions more than it does with sadness. Unlike the initial sessions where sadness was decoded more than neutral and happy emotions. As a final note, we'd like to highlight that in this study we have introduced a new solution for psychiatrists to treat their patients through regulating their brain activity using EEG-Neurofeedback techniques. For future work and development, we're planning on linking a virtual reality application with our brain-computer interface system for decoding and modulating human emotions. Connecting the system with the VR will make it more interactive and enjoyable. Moreover, we can create a collaboration with psychiatric clinics to implement the solution with patients who suffer from mental health problems and follow up with the improvement in their condition after multiple neurofeedback sessions, just to verify the effectiveness of the solution. And that concludes our presentation.